Good morning, everybody. We'd like to get started with the Grand Round season, 2019-2020 Grand Round season. It's fantastic to be here with all of you today. Great to get started, and we have a wonderful Grand Rounds presentation in a bit, and Joe Galerner will be introducing Renato, our Grand Round speaker today. I wanted to make a few comments. And I'm going to start out by getting the bad part of the announcements out of the way first, and then go to the exciting, good, happy parts. The bad part of the announcement has to do with over the summer, a university report was issued that described in detail allegations made against a former professor in the Department of Psychiatry, Gene Redmond, who was found to have both coerced and sexually harassed undergraduates in his research station in St. Kitts in the West Indies. Some of you may have read those articles. There were articles in the newspaper about it. Some of you may have read the report. That report is available, and we, meaning the executive committee of the department, will be sending out an email to people describing that event, and also providing links for support for people who feel are upset by the news or by the implications of that news. But it's a reminder about the vulnerability in any community for sexual misconduct and the terrible cost of that, both for the victims and the perpetrator. And you know, we do everything that we can in a community like ours to make sure that that sort of thing doesn't happen. But it's obviously distressing to think that something like that can happen. Let's all work to make sure that it doesn't happen again. I'm not sure that there's anything more that I can say. It's hard to move on to anything else after sharing such distressing news. It's probably old news for some of you, but I think it's really important to acknowledge the reality of these kinds of things, because if we don't talk about them, we can pretend like they don't happen, and that is not a healthy thing for a community. So let me shift to the happiest of happy news, which is that also this summer we welcomed our new residency class to Yale. They're exceptional people. We're thrilled to have them part of our community. Also our new class of psych interns and some new members of our faculty. Also part of the summer celebration was the news of 2 new endowed professorships in the department. These are 2 new recipients of the Elizabeth Mears and Howes Jameson professorships. One of them is Professor Joy Hirsch, and the other one is Stephanie O'Malley, Professor Stephanie O'Malley. And that's really just wonderful news as well. And I wanted to say 2 things about Grand Rounds, because I think that Grand Rounds are really an extremely important part of our community. The first is that we've had the opportunity to consider setting up distributed video recording, or whatever called tele-presentation of Grand Rounds, so that you could stay in your office all by yourself and watch Grand Rounds on your computer and interact with nobody. And I'm going to suggest that the point of Grand Rounds, that undermines the actual deep deep value of Grand Rounds in our community. That the deep value of Grand Rounds is getting people together all in the same room, interacting, getting to know everybody, and importantly, to interact with people who don't do the same kind of work that you do, who don't have the same kind of background that you do, who approach this kind of work from a completely different and other perspective, and to all come into the room and see how this mix, what comes out of the mix. What often comes out of the mix are friendships and find people that you like to interact with, and people that you never met before. You meet new people, and that's really valuable. And those connections that are made in Grand Rounds are really, in some ways, part of the substance of our department. And I'd like to really encourage people to take advantage of this, because the more people who come to Grand Rounds, the more valuable Grand Rounds becomes, the more lively, the more exciting, and the stronger our community as a whole becomes. So I'd like you to really think about Grand Rounds as more than just the topic of the lecture of the day, but really the opportunity for us to get together as a community and work on getting to know each other, and only good things come from that. The second thing about Grand Rounds is that I'm supposed to tell you about CME credits, and that is that if you want CME credit, we no longer, the faculty, we no longer have sign-up sheets. You should have, the site is psychiatrygr@yale.edu, and if you go to that site, you can get credit for your CME credit for Grand Rounds, and encourage you to do that. So, oh yeah, I was going to tell you what's coming up, but that's on the other side of the page, and I had to remember to turn over the page. So next week, we'll have a lecture by George Dragoy, who's a faculty member in our department, called Neural Ensembles, Underlying Internally Generated Representations. And George does work on a part of the brain called hippocampus, and how the hippocampus makes maps of the spatial world around us. It's really innovative and exciting work, and I commend that to you. And then the following week, which is the 20th of September, Ariel Baskin-Summers from the Department of Psychology at Yale will be talking about understanding disinhibition from diagnosis to mechanism to treatment. She's a very creative cognitive neuroscientist, psychologist, who studies impulsive behavior, sociopathy, things like that. And she's a really great speaker. She spoke at the Community Neuroscience Conference last year. So those are the upcoming Grand Rounds lectures, which will follow the exceptional lecture that you're going to hear today. And I'd like to invite Dr. Glanter up to the podium to introduce the speaker.
So it's really a great pleasure for me to introduce Dr. Renato Polimanti. He trained BS and master's and PhD, PhD in evolutionary biology at University of Rome, Tor Vergata. He came to my group for a postdoc starting in 2013. He was created an assistant professor in our department in 2017. He's done really exceptionally well across the board. He's done very well with grants, with 2 NIH R21s, 5 private foundation grants, including a NARSAD, multiple awards and honors, including a Simons Foundation Autism Research Initiative Explorer Award, and a Young Investigator Award from the American Foundation for Suicide Prevention. He holds several editorial board positions, and he's also co-editor with me in the journal Molecular Neuropsychiatry. We're just taking over. We need to encourage submissions. If you have anything good, send it to us. We want to raise the impact factor from imperceptible to, something perceptible. He is already the author of 84 peer-reviewed papers and about a half dozen more available on Bioarchive, some of which are going to be really, really big. He's published in excellent journals, Biological Psychiatry, Molecular Psychiatry, Nature Neuroscience. Renato is a sophisticated statistical geneticist. He's a key analyst both for the Million Veteran Program and for the Psychiatric Genomics Consortium, SUD, Substance Use Disorder Group. He's made major contributions to numerous MVP papers and is lead author on the new PGC opioid use disorder article, which is about to be submitted. And he's not even going to have time to talk about that one today, but it's a really good paper. He's a really outstanding young scientist, and he's brought a lot to the psychiatric genetics research program at Yale, and he will bring a lot more. So it's my pleasure to introduce Renato.
Thank you for the introduction. It was really, really nice, maybe too nice. So I'm going to talk about big data, why we need big data in psychiatry genetics, and so why we are using consortia, biobanks, citizen science to increase this sample size, and why we need these huge samples. And if you want to take pictures, if you want to tweet, there is my address. And so do it or don't do it is the same. I'm not that social, so it's fine. So I have not conflict of interest, at least financial one. However, I have several academic interests. Human genetics is my first, is my love. Obviously, biological psychiatry. I like really a lot evolutionary biology, statistics, and computational biology. So I try to mix up these things because of the complexity of the issues that we have to investigate. So my ultimate scientific goal is to link human genome and human phenome from different perspectives. And the reason is that the human genome is affected and shaped by multiple forces. So on your right, we have few terms from population genetics: a human demographic history, genetic drift, natural selection, sexual selection. All these forces shaped the genetic architecture of human genome. And they shaped also the predisposition to Mendelian phenotypes and complex traits. So in few slides, I will explain the difference between a Mendelian trait and a complex trait. But first, when you start to do some genetic research, this is the first thing that you notice. And these are few figures from the Human Genome Diversity Project and the 1000 Genome Project. These are not really recent, but the impact of these studies is outstanding in the human genetic field. So on your right, you can see a phylogenetic tree of the Human Genome Diversity Project, which include 54 human population, and you can see that there are clusters, and each cluster is a continental group. So you have Africa, Middle Eastern, Europe, Central Asia, East Asia, Amerindians, and Oceanic. So we have this cluster. And then from the Human Genome Diversity Project, we have a map, 24 population, and here we can see different pies, and the dark slice, the gray dark slices are the ones that are variants that are present in multiple continental groups, in multiple population. And then we have the colored one. They are the variants that are specific for a continental group or for a population. So we have some common variation and some ancestor-specific variation. And then here on the bottom, we have the number of variants in a human genome, so around 4 million, 4.2 million variants, if you are not from an African population. Indeed, you can see here European, East Asian, Central Asian, a mixed American population are about the same amount, 4 million variants. And then you have the African population. So that reach almost 5 million variants. And there is a reason for that. Humans are an African species, so there was an out of Africa, a bottleneck, a reduction in the diversity. So non-African individuals are the outcome of this out of Africa. So there is this bottleneck and we have less variants. So when we talk about variants, the main difference is between rare and common variants, and again, the 1,000 genomes data. And here you can see the frequency, the alternative frequencies, because the easiest example is a biallelic variant, so one variant with two alleles. So you can see that the highest number of variants are rare, more than 10 million variants, and common variants are not that frequent. So most of our genome is affected by rare variants. And here you have the number of rare variants detected in the 1,000 genomes population, and again, these are the African population, and you have a higher number of rare variants in the African genomes. Here, the colored one are the singletons, variants that are detected only in one individual. So, and this number is the same across all human populations. So, coming back, so we have the genes, we have variants, and what these things do, they are linked to phenotypic expression. So we can distinguish between Mendelian and multifactorial inheritance. So in a Mendelian phenotype, you have a single gene, a single mutation that is actually causing the phenotype. Is 100% variants explain. You can have modifiers, genes that are not causing the phenotype. The phenotype is caused by the mutation, the causal mutation. But they can change the genotype-phenotype correlation. So you can have individuals with the same mutation and sometimes slightly different phenotypes. And then we have the complex traits. We don't have a single causal variant. You have multiple susceptibility genes. And the next slide I will show how many they are. And the combined effect of these genes together with the environmental risk factor lead to the onset of complex traits. So, what about complex trait, which will be the focus of my talk today? As I said, multiple genetic factors. We have also an environmental component, so we can split these variants between the one due to the genetic component, the heritability, and the one due to environmental component. And one easy way to do it, almost easy, is twin studies. So determining the difference between identical twin and fraternal twin, we can estimate the heritability. And we saw that across complex traits, there are traits with a higher heritability and traits with a lower heritability. Here you have like different psychiatric disorders, and you can go from 80%, almost 90% for bipolar schizophrenia to around 30% for generalized anxiety disorder. So all these psychiatric disorders have heritability. There is a genetic component. But what is the difference? So let's come back to rare and common variants. And here I put an example that is autism spectrum disorder. I choose this example because I work on this thing and I was writing a grant until a few days ago, so I had this figure ready. But there's also another reason. Like if you think about autism spectrum disorder, you think about rare variants. And they taught us a lot about the biology of this disorder. Here you have a list of all genes affected by rare variants, de novo or inherited variants, and different syndromic forms. And if you go to an autism, like when I go to the Simons Foundation meetings, 80% of the talk is about rare variants. I don't work on rare variants. They are nice, but I like common variants better. So, and if you see here, common variants explain 49% of autism spectrum disorder. So rare variants are important. They are easy to understand because you have one gene affect the mechanism, and you can create, for example, an animal model, and then from there try to understand the biology of the disease. Common variants, they are much more complex, especially because the effect is really small, and I will tell you how small it is. So polygenicity. Here, another example, schizophrenia, and this is the lifetime risk of schizophrenia, depending on the relationship with an affected patient. So you have a monozygotic twin, and you share 100% of your genome with this monozygotic twin, and your risk to develop schizophrenia is 48%. But then you can have like dizygotic twins, children, sibling, parents, and you share 50% of the genome. And your risk goes from 17 to 6%. And then maybe you are a second degree relative, and your risk goes from 6 to 2%. Or maybe you are a third degree relative, your risk is 2%. Or you are unrelated to the patient, so your the general risk in the population is 1%. This means that we don't have probably a schizophrenia gene, but there are multiple genes. How many? We don't know. We have estimates. I will show how many they could be. So if you have multiple genes, this is the risk stratified. If you have one variant, so you have a higher risk. If you have 2 copies of the variant, if you have 2 causal genes, 4 copies is the highest group of people with risk. 3 SNPs, 10 SNPs, 100 SNP. Increasing the number, you start to have a bell-shaped curve, and you will have on the tail, you will have on the tail people with high risk. They carry a lot of risk variants. So we know that common variants are associated with psychiatric disorder. They explain a lot of the phenotypic variants. There are many variants. We have to find the variants. So there are different approaches, and they were developed depending on the technology that was available at the time. So initially we had family linkage study. They were conducted on informative families, so families that have multiple cases. And then at the time we were able to investigate informative marker across the genome. They wasn't really dense, so we had few informative informative markers. And from that we can identify shared region across these family members, and from that arrive to the mutation. And from the mutation we can have an hypothesis. This kind of approach works really well if you have a large effect size. I will talk later about large effect size, but doesn't work that well for common variants. So another approach is maybe you have an hypothesis about what is the cause of your disease. And among these hypotheses, you find a gene that you really like, and this is the gene that is doing that is causing this phenotype. And within that gene, you have one variant, and it's the variant that maybe is associated with a protein change, and you think, okay, that's it. I'm going to do my candidate gene association studies comparing cases and controls, and I want to see if the cases have higher frequencies of this deleterious variant. Unfortunately, it didn't work that well because there is a problem. We are really bad to make an hypothesis, and I will explain in the next slide why we are really bad, but there are a few exceptions. So then we have genome-wide association studies. These are hypothesis-free. We don't have any hypothesis. We test the entire genome. We need large sample size. I will talk later about how large. We find a SNP, then a common variant, a gene, and from that, an hypothesis. So let's talk briefly about candidate gene studies. Everyone has his favorite gene. I have my favorite gene. It's the one that I started like almost 10 years ago when I was doing my master thesis. But we now know that most of these candidate genes, they are not really relevant for the phenotype. And these 2 papers, they analyze schizophrenia candidate genes and depression candidate genes, and they found in most of the cases there is no support. There is no evidence that they were real signal. Unfortunately, still a lot of people are doing candidate gene association studies, and you shouldn't do it. However, we have few exceptions, like for example, ApoE in Alzheimer research, or alcohol metabolism gene in alcohol research. These were detected in candidate gene association studies, and they're replicated in GWAS, because if the variant is really important, you should find it also in a GWAS study. So before to start a candidate gene approach, take a look to the false candidate genes hypothesis that we did in the past. We have to learn from that, because these issues caused a replication crisis in human genetics. So because when we were starting to do association studies, we were saying, okay, we got the gene, this is it. And then no one was able to replicate it because the sample size was too small and the gene was not really relevant. So we moved from there when the technology permitted us to investigate thousands, hundreds of thousands, and now millions of variants across the genome to do a genome-wide association studies. Genome-wide association studies, GWAS, are not elegant experiments. This is a brute force experiment. I'm testing everything because I have no idea what's going on. So we use arrays that can genotype millions of variants and then impute other variants. So variants that we didn't genotype, but through a reference panel from sequencing data, we can predict other variants due to their linkage equilibrium, the correlation that we have among these variants. So right now, you can have a GWAS plus imputation that arrive to more than 10 million variants, 15 million variants. So this is a lot of information, and there is a lot of way you can screw things up. So you have to be really careful because it's a lot of power. When you use these brute force experiments, you have to be really careful and you have to know what you are doing. So what kind of variants we can detect with a GWAS? There is a relationship between allele frequency and effect size. How much risk is associated with that variant? So when you have a Mendelian disorder, you are here. The variant is really rare, the effect is huge. In some cases, if it is a Mendelian trait, explain completely the phenotypic variants. And then you can have few exceptions, as I said, ApoE, the CFH variant that is associated with the age, macular degeneration, and other variants, and these are exceptions. So when we did the first GWAS, we found these variants only because we're conducting small studies. And then from that, increasing the sample size, we started to find a lot of variants that were common here with tiny effect size. So this what means that first, complex traits are highly polygenic. Then we need huge sample size, big data to crack the genetics of this complex trait. And each complex trait is different. They don't work in the same way. So in this slide, we have 4 traits: inflammatory bowel disease, height, schizophrenia, and bipolar disorder. So you can see that for IBD, when they started to do GWAS on IBD, they were super happy because they were doing like a GWAS with 5,000 subjects, and then with 5,000 subjects, they got 100 genome-wide significant loci. A genome-wide significant loci is are variants that survive multiple testing correction for the entire genome. So they were super lucky, we did it, we got it, that's good. But for other traits, it didn't work that well. For example, for height, it was working fine here. When we started, we didn't know how large should be this sample. So 5,000 looked like this is a big number. We now know that 5,000 is a tiny number. So for height, we started to do like 10,000 cases, and we got a lot of variants, and then increasing it went better. For psychiatric disorder, we were not that lucky because psychiatric disorder needs much higher sample size. You can see here the dead zone, the zone when you have odd samples and you don't find anything. It's pretty bad for schizophrenia and bipolar disorder, and then it comes up when you reach a certain point. And that point is the inflection point that you reach when you start to have the power to detect the tiny effect sizes that are associated with that trait. And we predicted at a certain point we should reach a plateau where increasing the sample size, we will not detect anything. We found everything that we need. So this is the reason why we need large sample size and where we can get these samples. One approach is to bring data together. So in psychiatric genetics, at a certain point, they start to think, we are not finding anything by ourselves. We have to bring the data together and create a consortium. So this thing, I think, started more than 10 years ago and was started by Patrick Sullivan from UNC. And when he started to make these talks about we have to come together, bring data together, analyze data in the same way, people were not really happy about that. But at the end, he was able to found the Psychiatric Genomic Consortium. Right now, we have more than 1,000 investigators across the world, and is the largest consortium in psychiatry. And the aim is to reveal fundamental biology, improve clinical practice, and find therapeutic targets. So we are in the 3rd funding iteration of PGC, and the goal is to reach 100,000 cases per work group. And right now, we achieved that goal for depression and schizophrenia. And we are not there for the other, because we have different work group, ADHD, autism, bipolar, depression, and there is substance use disorder group. So we are not that yet. But we have also other consortia, and for psychiatry here is a list of different consortia, and there are other relevant for psychiatry, for example, the anxiety neurogenetic study, the Enigma study about brain imaging, there is the Social Science Genetic Association Consortium, and the Tobacco and Genetic Consortium. And there are other consortia for other traits. So this is one approach. You have studies that were collected and recruited for a specific a cluster of disease, and you bring the data together. Another approach is Biobank. So I'm talking about National Biobank. They are the largest one, and the first one is the UK Biobank, and it's not the first, but it's the one that made the largest impact on human genetic research and in human research in general, because first of all, they recruited 500,000 people. middle age, more than 4,000 phenotype assessed, brain imaging phenotype, they are going to release so body imaging phenotype. But the nicest thing that they did, they opened the biobank to everyone. Right now, any scientist around the world can apply to get the data, pay a small fee, around $5,000, maybe less if you don't need like all the data, and you can get the data. You can do your own analysis. So there are, I think right now, thousands of scientists working on this data, and there are a lot of papers coming out. So these data sources generate a huge amount of information. So another biobank, for example, is the Million Veteran Program. Right now, we have more than 750,000 veterans recruited. Is across the US. We have different centers, and it focuses on multiple projects. And the 4 flag projects that you can see on the website are about opioid, PTSD, cancer risk, and cardiovascular disease. And Joel, together other PIs in the VA, are leading both the opioid and the PTSD analysis. So how is different the MVP from the UK Biobank? MVP is not open to everyone. To access MVP data, you have to be a VA investigator, and then you need to get funded through an MVP mechanism, and then you can get access. So the access is still limited, and is a working in progress. So it's making an impact, but it's not fast like the UK Biobank, because there are not that many investigators working right now on this data. And then we have the US version of the UK Biobank, the All of Us research program. The goal is to recruit more than 1 million individuals across the US. And right now there is a website you can go with a browser and you can check the data. They have information about more than 100,000 individuals. And is going to make a huge impact, because the difference between this biobank and the UK Biobank is that this biobank reflects the genetic diversity of the US population. Like the UK Biobank is around 95% European descent. This biobank is much more diverse, so it's going to contribute more to understand differences across ethnic groups. And then we have also other kind of biobanks, much smaller, like institution biobanks, for example, Vanderbilt, Harvard, and there is also in process the Yale Biobank, with the goal to recruit more than 100,000 subjects and do whole exome sequencing on the subjects and bring the data to the clinical practice. So what else? Citizen science. And this is another way that scientists are getting data. So right now there are companies, and you can send them your saliva, and they will give you some information. The most known is 23andMe. Here is my stuff. And it's surprising I'm Italian, as you can hear from my thick accent. But I have also a 7% of Middle Eastern ancestry that I didn't know. And they can provide also information about how old was your connection with this other ancestral group. So other companies that can provide similar evidence about like Ancestry.com, but the things about 23andMe, and they did this from the start, is that they want to provide healthcare information. So you can have information about predisposition, like your ApoE genotype, other variants with large effect, carrier status for a lot of variants that can be associated with rare and Mendelian traits. So you have a lot of information. If you heard about the story of 23andMe, they started with the healthcare information, and then when stopped by the FDA, and a couple of years again, and they got back with the information. And there is a big discussion of how reliable are these data, because, for example, in some cases, there are stories about people that got positive results for a rare variant with a large effect, and then they went to a clinical laboratory, and the genotype was not confirmed. So there are some issues about how reliable are these data, but right now there are more than 10 million customers for 23andMe, and this is a huge amount of data that scientists are using, because you can fill a questionnaire online and people can use this data. You can say, okay, I want to participate to research, and your data will be shared with the 23andMe collaborators, mostly like academics group and pharmaceutical companies. So there is also some points to discuss, but this is not the place. So what's the difference about consortia, biobanks, and genetic testing company? So the first, in a consortia, usually samples are recruited with a specific focus, a group of disease, a specific disease. So usually you have a deep phenotypic characterization. But the opposite is they were collected by different groups. So sometimes they use different instruments. So there is heterogeneity in the assessment. So it takes a lot of time to harmonize this phenotype. And then you have the biobank. You have a large sample, like when it's a national biobank, UK Biobank, MVP, all of us, but is representative of the general population. So when you have rare or uncommon disorders, this sample can be larger, like 1 million subjects, but still you will have a low prevalence of this disorder, so we will be not power to detect, to investigate this kind of disease. And then we have seed and sign, genetic testing, massive sample size. But the phenotypic characterization is really low because mostly it's an online questionnaire, so it's not really deep. Like when they did the first depression GWAS on 23andMe data, there were 3 questions. And it worked because the sample size was huge, but still there is a big question. There are a couple of papers, one from Jonathan Flynn and Ken Kendler about how the phenotypic heterogeneity is affecting your genetic results, and there is an ongoing discussion. So let's talk about psychiatric disorder and GWAS and how the sample size can impact your result. So here are 2 GWAS from PGC, the Psychiatric Genomic Consortium, 2011, about 10,000 cases, 12,000 controls. They found 5 genome-wide significant loci, so variants that survived the bone ferroni correction accounting for the entire genome. And then, like 3 years later, 37,000 cases, more than 100,000 controls, they got more than 100 genome-wide significant loci. This is only power. This is a brute force experiment. They found a lot of genes, and there was a huge impact. I remember this was 2014. It was my first SOBP meeting, and the SOBP is not a meeting focused on psychiatric genetics, but I saw this Manhattan plot many times because there were people investigating each of these variants and how they were affecting schizophrenia disorder. So how to read a Manhattan plot? Here you have chromosomes, so you have 22 or plus the sexual chromosomes, and here you have the negative log 10 p-value. So higher is your peak, strongest is the association, each dot is a variant. So sometimes you have a column, and this is due to the linkage disequilibrium, the correlation among the variants. So when I'm saying 100 genome-wide significant loci, I'm saying 100 independent signal across the genome. And this is the effect for major depression. So 2013, 10,000 cases, 10,000 control, nothing. And then this year, a huge amount of data. 250,000 cases, more than 500,000 controls. They found 102 genome-wide significant loci. If you compare these 2 traits, depression and schizophrenia, you can notice a difference. Like with a much larger sample size, they got the same amount of genome-wide significant loci. So this means maybe that depression is more polygenic. So I'll talk about this later. So there was a time back in 2012, before we reached this large sample size, that funding agencies were almost shutting down PGC because, as I said, we had the dead zone that was really deep for psychiatric disorder. So they were saying GWAS is not the right approach for psychiatry. So there was the letter from Patrick Sullivan and the other 96 PIs in PGC, don't give up on GWAS, and they were right because we were close. We needed only more samples. And why is that? This is due, as I said, to the fact that we have small effect size. Yes, you can see here. So here are 2 density plots for continuous trait and disease. Here you have the effect size. Here you have the density. And you can see here that you have 2 peaks. One is from neuroticism, and this means that the effect size are really tiny. Here most of the variants are in this place, and the other one here is depression, the one in the light blue dotted line. This means, and explain why we get this effect, why we need a much larger sample to get 100 genome-wide signal loss, because the effect size are much smaller. So there are different hypotheses to explain this effect. One is the heterogeneity, like depression is a much more heterogeneous phenotype than schizophrenia. So we may have multiple subphenotypes and with different genetic architecture. The other is that evolutionary pressure. Like I said, there are multiple aspects that interact together. So purifying selection is a mechanism by which selective pressure remove variants with a deleterious effect from the population. So there are certain genes, in particular brain genes, that have a strong evolutionary pressure. This means that variants with large effect are removed. So they have a really low frequency in the human population. So this effect is may be particularly strong in depression, but is present across all psychiatric disorders. So here is a different kind of Manhattan plot. You have chromosomes here. Here you don't have p-values. Here you have heritability. So higher is the peak, the more heritability you have in that specific region. And we have here 3 traits: dyslipidemia, hypertension, and schizophrenia. And here you can see we have high, really high peak that explain up to 1% of the phenotypic variants. For example, chromosome 19, where is the APOE gene, but you don't have any high peak for schizophrenia. And then in this study, they did like a simulation analysis. This is the percentage of the genome where you have a 0 heritability estimate, and it's really low for schizophrenia. And to explain this low portion of the genome without any kind of heritability, for schizophrenia you need 20,000 variants. This means you have 20,000 causal variants with tiny effect for schizophrenia, about 10,000 variants for hypertension, and 2,000 variants for dyslipidemia. So this means that when you do a GWAS, for these 2 traits, you need a much smaller sample size to get these variants. Instead, with 25,000 with tiny effects, you need a much larger sample size. So polygenic, that's it. No, we want things even more complex than that. So polygenic or omnigenic. Like a couple of years ago, he came out from Jonathan Pritchard at Stanford, and he came out with this idea. Maybe it's not polygenic, maybe it's omnigenic. Omnigenic means that it's not 20,000 variants. It's the entire genome that is affecting the predisposition to complex trait. And he came out with this data that was published in Cell this year about the relationship of gene expression. So use gene expression data to explain this hypothesis. And the hypothesis is that you have some core genes that are directly affecting the phenotype that you are interested, and then you have all the peripheral genes. They are linked to the core genes, and they are affecting the core genes, and they will also be associated with your phenotype via this indirect association. So polygenic, omnigenic, that's really complex, and GWAS are helping us, and now we have like thousands of genetic association. These are a plot that is really well known in genetics from the GWAS catalog, where there are the list of all the genes and variants identified. You have the 22 chromosome plus the 2 sex chromosome, and each dot is a genetic association. Each color is a different domain, and here you can see that it's everywhere. It's highly polygenic. So RG was great, depending on the day. So missing heritability is the big issue for GWAS, because it's the mismatch between the twin study and the GWAS results. So I showed you before that for psychiatric disorders, we have like from 80 to 30% of variants explained by genetic factors. And then we have 2 cases of missing heritability. The first is the old case. So you do your schizophrenia GWAS. Schizophrenia has a 80% heritability, and then you get only 5 variants from that. And you say, where is the heritability? I should have a larger number of variants, or at least these 5 should explain higher variants. Instead, they explain very, very little. So the first case is explained by we are not really powered. We were not really powered to detect the variants and the tiny effects. But we have also a recent case. So we have this huge amount of data, hundreds of variants, but still the heritability estimates that we have from the GWAS is much lower from the one that we observed from twin study. So schizophrenia, 80% from twin study, GWAS of schizophrenia, 20%, 25%, so we are missing a big chunk. But this year came out a paper on bioRxiv, it's still a preprint, so they found the missing heritability. As I said, when you have a good GWAS plus imputation, you can reach up to 20 million variants, and these are heritability estimates. I'm not going in the details here for height and BMI, And here you can see with height, you have an heritability between around 60%, for BMI between 25 and 30%. And then with the whole genome sequencing, so you are sequencing the subjects. You don't find only the variants that you have the probes to, but you find all the variants. You reach a much higher number of variants, 47 million variants. And they calculated the heritability, and they found an increase. So for height, between 75 and 80%. For BMI, I'm reading 40%. And so there is a chunk of variants that we are not able to investigate with the standard GWAS approach, and are the variants with the low LD here, variants that are not correlated with the other variants that we cannot impute, and they are bringing the misheritability. So this worked for height and BMI, and this trait worked really fine with GWAS. We don't know if these things is going to work for psychiatric disorders or other complex traits. So why you are not doing whole genome sequencing if they are better than GWAS? Because whole genome sequencing are much more expensive than GWAS. Right now, to do a GWAS in one subject, you spend around $100. For doing a whole genome sequencing in an individual is about $1,000. So since you still need the huge sample size, because the effect size are still really small, we don't have right now the possibility to conduct whole genome sequencing on a huge sample size. So it's coming. Let's hope the price will go down, or like Consortium, people bringing whole genome sequencing data together. So what we can do in the meantime with the GWAS data? One approach is polygenic risk score. So here we have our Manhattan plot, and this was a really good GWAS because we have a lot of variants that survive the line, the Genome-Wide significant threshold. So we have this small effect size, a list of variants, so we can create a score that sum the effect of each variant and the number of risk variants that you have in each individual, and so you can calculate a score for each subject in your cohort. And there is a really nice article in Biological Psychiatry about a review article about polygenic risk score in psychiatric disorders. So there are different methods that you can calculate polygenic risk score. So this sum is the easy example, and there are different thresholds that you can consider. So at the end, you will have a number that you can put near your subjects. And what you can do with that? So there is a big ongoing discussion about, okay, we have this polygenic risk score. We can stratify our population for genetic risk, and maybe we can tell to people with the high risk, you have a high predisposition to develop these disorders. And some other people say, no, polygenic risk score works, but in research. You have a lot of issues like population stratification, differences across, and pleiotropy also, because sometimes your polygenic risk score is not predicting your trait, it's predicting something else. So in last year came out this paper on Nature Genetics, and here you have your distribution, that you have your polygenic score in your cohort, and they did it for different complex traits, no psychiatric disorder, unfortunately, and they found that if you stratify your sample for the highest percentile, people with a high risk, genetic risk, you can have like 3-fold, 4-fold, 5-fold increase in risk depending on the threshold that you're considering. This is the example for coronary artery disease. You have the difference between control and cases, and then if you plot the percentile of your polygenic risk score distribution and the prevalence of the disease, you see that people with the highest, in the highest percentile, have a much higher prevalence of the disorder. So there is a big ongoing discussion about should we move polygenic risk score to clinical practice, especially because in some cases the polygenic risk score is predicting the same risk that you are now using for monogenic mutation. So there are monogenic mutation associated with like lipid levels that right now you are telling people you have this mutation, you have this familiarity, you should change your habits. But we can have something similar with polygenic risk score. So the problem is that for monogenic mutation is much easier because there is only one way to calculate. You have the variance, you don't have the variance. For polygenic score, there are thousands of ways to look this data, compare the lowest percentile to the top percentiles, so it's really complex. But people can right now access polygenic score. This is again my own data from 23andMe, and they provide polygenic score information about type 2 diabetes. So in the US, right now, there are 10 million people that can access this information. And they have a white paper on the website. It's not peer-reviewed. And if you see here, the predictive power of across different ancestry is not that good. And also the way they explain the polygenic score, your risk, is not that careful. They are saying that you are in the typical range. I have personally a 20% chance of developing type 2 diabetes between my age and 8 years old. They can provide the risk across different age group, but what this means. So we could have like people, like I work in genetics, so I know that this is not really reliable and doesn't mean really anything right now. But you can have people going to their clinician and saying, okay, I did 23andMe, and they are saying that I'm going to develop diabetes. And the clinician should know. Maybe he knows genetics, so he can explain, no, this is nothing. Or maybe he has no idea what genetic is. And especially because polygenic score is really an area that is not well known, for example, in clinical genetics, where they focus on mutation. So it depends on what kind of background each clinician has. So people have access, but there is also another project, the eMERGE, the Electronic Medical Records Genomics. We are in the 4th iteration. The previous one, they mostly focus on variants with large effect, how to get back to the patients and integrate genomic information with the health, with electronic health records. And in the 4th iteration, that is under construction right now, that should start in 2020 to 2025, they want to assess the genomic risk and how you can use the genomic risk to manage patients and to change your healthcare possibility. So if you see here, you have different centers that were involved in the eMERGE, and if you look carefully, there is not Yale. That's a shame. So we are going to change that. We are trying to. So we put together a proposal. There is Joel, there is me, there is John Crystal, and many other investigators, both from psychiatry and from other departments. And we want to try to do this genomic risk assessment in the Yale Health care system. So what about psychiatry? In our eMERGE proposal, we have a lot of psychiatric disorders, but there is also PsychEMERGE that is led by Jordan Smoller and Leah Davis. And they are trying to put together data from different biobanks, national biobanks. So there is Vanderbilt, there is Harvard, and put it together genetic information with the electronic health records. And this is the schizophrenia polygenic score, which is the most powerful right now in psychiatry. And you can see here the predictive power of schizophrenia with respect to schizophrenia in the electronic health record and other disorders and phenotypes. So you have psychosis, you have other schizophrenia-related disorders, you have PTSD, bipolar, suicide behaviors, and so on. But here you can see there are two ways to look the data. The first, the blue one, you compare the top decile with the bottom deciles of the score, and you can see the effect is much larger. And then you have the top decile of the score versus the remaining distribution. So which one are you going to use? Which one is the right one? That's a big problem if you want to bring the information back, if you want to stratify the population according to these scores. So this is a big issue, and probably with the eMERGE, we will be able to fix that, or at least trying to. In the meantime, in my group, we really love polygenic score, and we work also on autism spectrum disorder. We are curious because I said a lot of the variants in autism is explained by common variants, but a lot of people that don't have the disease, they have risk alleles for this disorder. So we did an analysis, and this is Frank, postdoc in my group, and we did the analysis using the PGC, the Psychiatric Genomic Consortium Autism GWAS. It is not that large, but the variants explained by the GWAS is higher than you expect for other traits. And we used the Philadelphia Neurodevelopment Cohort and investigated more than 500 phenotypes assessed in these children. And we found that in this particular middle age group, a significant result for the association between the autism polygenic score and the ability to recognize angry faces. And this was significant in the middle group and replicated in the adult group. And again, here we can do the same thing. We can stratify the risk across the population that we tested, and we can see that people that carry more autism risk allele, they have a much higher increase. In this case, not risk, but ability to recognize angry faces. This is a complex story, and we have different hypotheses, so if you want to read the full thing, there is a preprint on medRxiv, and you can read the whole analysis. But there are other things that we are interested in, and another thing is the genetic correlation. So we have this pleiotropic mechanism when one variant is associated with multiple traits. So this figure is from the Brainstorm Consortium, another consortium that is a consortium of consortia, because they brought together all GWAS from different brain-related disorders, so 25 brain disorders, 17 phenotypes, more than 1 million individuals, was a huge analysis, and they found genetic correlation. But what genetic correlation means? Genetic correlation means that you have a variant, as I said, that is associated with multiple phenotypes, but this could be due to different mechanisms. One mechanism is the vertical pleiotropy, when you have your variant that is associated with a first phenotype and then is associated with the second phenotype. So if you test the genetic correlation between these two, you find there is a genetic overlap, but it's actually due to the causal effect of P1 on P2. And then you can have an horizontal pleiotropy, where the variant G is associated with the two phenotypes independently. This means that there are independent mechanisms. There is not causal association between P1 and P2. So a big problem in epidemiology is to distinguish between association and causal effect. And the best way to do it is to do a randomized controlled trial and test the causal effect between phenotypes. But we can use genetic information to do a similar experiment. So a Mendelian randomization study, when we use genetic information to stratify our sample between people with high risk, for example, of low LDL cholesterol, and the other people, and for example, looking for cardiovascular events. This is the example. And you can find that using genetic information is useful because genetic information are not biased by environmental factor. So your cholesterol level is biased by your diet, your physical activity, and other environmental factor. Your genetic predisposition to develop a certain level of LDL cholesterol is not linked by the environmental factor. So changing your diet is not going to change your genes. You can change your level. So using this genetic instrument, you can remove the confounding factor of several environmental risk factors. So we did this analysis that I led from the Psychiatric Genomic Consortium. We tested the association between post-traumatic stress disorder and educational attainment. And we used different approach, different instruments related to a different disorder. And what we found is that there is a negative association between PST and educational attainment in this direction. So educational attainment is not causing PTSD, but PTSD is causing the reduction of the educational attainment. The data are available on JAMA Network Open. So we have a causal relationship, but we are not sure about which is the mechanism. Because one mechanism could be that educational attainment is associated with another mediator, and the mediator is actually causing the change in PTSD. So here we have that different variables can affect this association. For example, risk-taking behavior, trauma exposure, in this case childhood abuse, income could affect the relationship. So we did a multivariable Mendelian randomization where we combine these risk factors and see the independence of the effect. So the first model includes all the 4 factors, and here you can see that risk-taking behavior is not changing between the Mendelian randomization and the multivariable Mendelian randomization. Same thing for childhood abuse, same thing for income, but for educational attainment, we see a change. The univariate Mendelian randomization, we have the negative effect. When we adjust for these other factors, the causal effect is gone. There isn't anymore. There is something else to explain the association. So we did other 3 models combining educational attainment with the other moderators, and for risk-taking behavior, there is no difference. Childhood abuse, there is no difference. But then when we go to income, socioeconomic status, we see the big difference. So income, there is no change, and we see again educational attainment, univariate, there is negative association. Adjusted for income, the effect is gone. So our hypothesis is that the association between educational attainment and PTSD is mediated by income. And we did other analysis to try to understand what's the biology in the income GWAS, because we are talking about a GWAS of income. And we can see that there is a difference between income and educational attainment, where educational attainment is much more informative for brain-related mechanism, while income is not that informative. So if you want to read the full story, you can go to JAMA Network Open and read the entire paper. So we are still interested in educational attainment and socioeconomic status and their relationship with brain-related phenotypes. This is something that Frank is doing, and we are putting together a lot of information here about psychiatric disorder, externalizing behaviors, social science outcomes, personality traits, and brain volumes. And we are seeing that there are differences across psychiatric disorders. And we are doing Mendelian randomization, genomic structural equation modeling, latent causal variants to understand the mechanism and how these relationships are affecting our risk to develop psychiatric disorders and which are these mechanisms. So this is not published yet. We are working on, and we hopefully will post like a preprint in few weeks. But I really like Mendelian randomization and understanding genetic periodicity. So another nice thing to understand, for example, is finding biomarkers for trauma response. And again, you have a lot of environmental confounders when you do like an observational study in the blood biomarkers that you actually measure with respect to trauma response. You don't know which is the direction. You should do a longitudinal study with multiple time points, and this means that you need a large amount of resources because you need a large sample size, and sometimes you don't have it, so your sample is small and can be biased by other factors. So we used genetic information, and we tested 122 metabolites that were assessed in almost 25,000 subjects, and trauma response in the UK Biobank between 50,000 subjects and 100,000 subjects. And we did first a genetic correlation analysis, and we find that trauma response, different symptoms, avoidance, distant, being upset after being reminded of a trauma expose, being irritable, repeated thoughts about traumatic experience. So all these symptoms, they were assessed in the UK Biobank through a mental health questionnaire online. And so there is some overlap with the PTSD symptoms, although they are not exactly the same things. So we found some genetic correlation. We did our Mendelian randomization analysis, and we found four. There is one that has like probably also a component of horizontal pleiotropy, so shared mechanism. So in this case, we have like only the vertical pleiotropy, a causal direction, and direction is actually, these blood metabolites are actually affecting trauma response. We don't see the other way around, at least for these three. So it's the glycoprotein acetyls, the citrate, and the LDL cholesterol. So in two cases for GP, glycoproteins and citrate, we went in the literature and we found evidence in small study about the association of these metabolites with traits related to trauma response, like depression, anxiety. But these were small study, and this was cross-sectional studies, so they didn't have like the ability to distinguish between which is the direction. They only reported the association. Here we are showing with genetic information that there is a direction, and direction is this way. So we have like a discussion about which may be the mechanism. There is a preprint on bioarchive, so you can read it if you are interested, or asking me questions after the talk. But we did also another analysis, and we were interested about computerized device use and psychiatric disorders. So everyone is using a phone or playing with the phone or play video games, so they are widespread in the population. And there is a lot of discussion about which is the effect on mental health and on our behaviors. So we use data from the UK Biobank and the Psychiatric Genomic Consortium, and we did first our genetic correlation analysis. And we found correlation across multiple traits, and the strongest one was between ADHD and using the phone, in the amount of using the phone in the last 3 months, and playing video games and schizophrenia, a negative association. So we did again our Mendelian randomization study, and in this case we didn't find a causal direction. So we found that in both directions there was an association. This means that there is horizontal pleiotropy. There are mechanisms that are shared between these two pathways. And doing like a enrichment analysis and try to understand which is the biology, we found evidence of dopamine transport and the RD2 gene for the relationship, the negative relationship between schizophrenia and computer gaming, and FOXP2, the language gene, in a positive association between ADHD and phone use. And in this case, we were also able to do a sex stratify analysis, and we observed that this shared mechanism is much stronger in women. So we have a lot of hypotheses also about hormonal regulation and FOXP2 activity. So if you are interested, we have another preprint on bioRxiv that you can read. So these are the things that we are doing in my lab, and we are doing other stuff cool with MVP that I cannot show you because I didn't get the authorization. I was late. And PGC, the opioid GWAS, that we are almost done with that. But there are big challenges in human genetics. The first, as I said, large sample size, heterogeneity, the assessment. But probably the biggest challenge that we have, especially because seems that we are really close to make an impact on healthcare, is diversity. Here you have the GWAS reported in the GWAS catalog, and you have different ancestry group, so these blue things, European ancestry, and then you have East Asian, the pink, and then you have other African ancestry is the orange here, and so there is no diversity. And if you have to derive a polygenetic score from a GWAS done in European ancestry in another ancestry group, you have big issues. Less power, population stratification. So if we are making an impact in healthcare, this means that right now we are going to create health disparity across ethnic groups. And here is the analysis done by the PAGE study. That is an analysis about a US representative court, and you can see how much diversity it is. These are minority groups, so African American, Hispanics, Asian, Native Hawaiian, Native Americans, and you can see how much diversity there is and how much admixture there is. So we need to improve our ability to investigate this group, switch and focus on diversity because we need more information. So another thing is the genomic revolution in healthcare is coming or not. So I put this slide about the relationship between hype and progress, how things when come out and they are new, everyone is excited. And in genetics, we are really good to be excited about things. So I counted at least 4 waves and crash in genetics, at least 3. The fourth hopefully will not come. So the first one when we started with gene candidate association studies. So they were really tiny, 10 subjects, 10 cases, 10 controls. We found a gene, there was a nominal association, and we got the gene, we have it, that's it, we did it, it's over. We found it, we are going to predict everything. And then we were not able to replicate, and yes, it was not coming. So first crash. And then there was the Human Genome Project. We sequenced the first human genome, everyone was so excited, billions of dollars, it's coming. We did it, now we know everything. We are finding everything. So that's it, it's fine. And then it didn't change anything because we have all the information, now we have to understand it, and it's really complex. And then we have the first GWAS, like they were underpowered, and we have the missing heritability, but the first GWAS was in age-related macular degeneration, and they found the variance with a huge effect. As a large variance explained, and we say, okay, that's it, we got it. Now we are going GWAS for all these traits, and we are going to predict who is getting what. And instead, it didn't work, so it crashed again. So people outside genetics, they don't like geneticists because we get all the headlines and we get overexcited because genetics can have a huge power, potentially, if we are able to crack the mechanism. And now we have polygenetic score. So everyone is excited. Again, we can predict something. We can explain variants. And there is a big discussion of what we are seeing. So the variance is high. We have the diversity issue. We have the pleiotropy issue. So what you are predicting actually when you put together thousands of variants. And also how you want to calculate your polygenetic score, how you define the threshold. So there are a lot of questions. Also, for example, the eMERGE project, to me, like if someone asked to me, can you do these things? No, you cannot do it. I wrote an entire grant about that, and I was saying, let's try this, but it's going to be really, really, really tough. It's kind of a moonshot, because right now we are not ready for prime time. Let's hope that when emerge start, we will be able to discuss with others and bring together that we will need maybe more than 1 year to validate the polygenetic score, because right now is 1 year validation, and then 4 years you go and you give the results to the people. So I'm not really sure about that. So are clinicians ready for polygenic medicine? My impression, it depends. It depends on which institution it is. It depends on how strong is the relationship with people that do polygenetic score analysis and people that do clinical practice. If they are the same people or if they interact a lot, yes, they may have the expertise. If there isn't this connection, people have no idea what a polygenetic score is. And this is my impression also talking with people that work in different fields. They are scientists, super smart people, but then polygenetic score, what is a polygenetic score? What a polygenetic score does, how you calculate it, thousands of variants, how you select the variants. So there are a lot of questions. It's not easy. It's definitely, I don't have a solution right now. The thing is that maybe we should introduce like classes about this stuff, like explaining what is a polygenetic score, explaining what is polygenicity, that yes, you have mutation, but most of the variants of human phenotype and disease is explained by tiny effects. So maybe we should improve that. So acknowledge Frank, Carolina, that I didn't mention, but you saw the picture, that she did some analysis, and Gita, that just joined my group. Obviously, all the funding agency, they gave us money to do some cool stuff, and let's hope to do it, really. And then all the other people in the division of human genetics, Joel, and all the other people that are mostly here. So thank you for the attention.